So, good morning, everybody. Depending on what time you are in, it's 9:20, and we will start this BroilerNet webinar now. I'm Stefan Gunnarsson, and I have the privilege to be the coordinator of the BroilerNet project. And this webinar has been planned by Natalia Mayo Masferrer from IRTA and University of Barcelona. And I will start with sharing my presentation and give a short overview of the BroilerNet project before we start the presentation by Dr. Arne Jung and Dr. Zoi Prentza. So the webinar today is facing the challenge Enterococcus infections in modern broiler production. And the program today is a presentation by Dr. Arne Jung from Veterinary College in Hanover, Germany, in understanding Enterococcus infections in broilers. And then we will have a short break. And then Dr. Zoi Prentza from PINDOS in Greece, which is a partner of the BroilerNet project, will be talking about advanced biosecurity measures in Enterococcus control. And then we will have a question and answer session. And during the presentations, you are able to put in questions in the question and answer function within the Zoom, and you will find it on either it depends on how you have designed your Zoom or if you're on the web version, but you will find it there, and then we will bring the questions to the presenters. So a brief presentation about the BroilerNet project. It's a European network project for exchanging on broiler production and exchanging knowledge. And the project has been running for 4 years and it's very soon to be finished. So this is the final webinar of the project. And we have been collaborating for 4 years. It's 25 partners in 13 countries. So it's from, I will have a laser pointer here, from Finland to Portugal and from Greece to Ireland. And in the countries, we have one academic partner and one industry partner. And the funding has been working that we are equal. So it's not in kind financing requested by the industry partners. Our work has been to build networks on national level. So we have connecting farmers and advisors on national levels in all the countries. And then on the international level, on the European level, we have connected these facilitators of the networks, and we have also created thematic expert working groups that have been evaluating. So what we have been aiming for is to fostering the interaction between science and practice, because a lot of scientific knowledge has not really been reaching the farmers. And by our way of working, we have asked farmers and advisors in the different countries what problems, what challenges they have. And then we have tried to find good practices that, for example, country A consider that they have a challenge with a problem on the broiler farms. And then we find good practices that have been applied in other countries. So it is to distribute knowledge freely and openly. And then in order to make this comprehensive, we have organized this in animal health, and there Natalia Meyer from IRTA have been the coordinator for the animal health management theme. We have animal welfare, where the group from Friedrich Loeffler Institute in Celle has been coordinating the animal welfare questions, and the environmental sustainability have been coordinated by CRPA in Italy. And this is not a way to have closed borders between things, because animal welfare and animal health is very closely linked to each other. But it has been a way to structure the work. And I would also like to say that the work has been focusing on what's happening on the broiler farm. So the focus on the hatcheries and the abattoirs, it's not within this project. The focus has been the farm, the farmer, and the broilers on the farm. That is the main focus. And we have a lot of information. I will put these links in the chat later on. And you can go into our BroilerNet Knowledge Hub to find information. We have more than 100 good practices. We have dozens of videos, animations, and we also have videos of the webinars we have had. And as you see, the webinar of today is also recorded and will be available in a couple of weeks at the BroilerNet Knowledge Hub. So thank you very much. And here are all our partners from the different countries. I will stop sharing now. And I would like to welcome one of our experts, and that is Arne Jung, which is has a PhD, and he's also a Doctor of Veterinary Medicine Habilitatis, and he's a expert in microbiology and in poultry, and he's also a European specialist in poultry veterinary science. And he's working at the Stiftung Tierärztliche Hochschule Hannover, the Veterinary College of Hannover. So please, Arne, you're welcome.
Yes, thank you for inviting me today. So I was asked to talk about Enterococcus infections in broilers. And I will start with an outline about my talk today. So I will give an overview over the different Enterococcus species involved in disease. Then I will mainly focus on my main research topic. So this is Enterococcus cecorum, the importance in the broiler production, pathogenesis, and the clinical presentation and pathology. And then at the end, I will give a short overview about the intervention methods, which can be discussed later, maybe. So, as you may know, so the Enterococcus belong to the order Lactobacillus, and it is, the origin of the name is from the Greek, so enteron and coccus. So it's easy to remember this, and it's a gram-positive ovoid coccus. And now around 73 species are published, but not all of these can be isolated from poultry. Just a few. So mainly they are commensals of the gastrointestinal tract of humans and other animals, but they can also be found in water, in soil, plants, or in fermented foodstuffs. Also, it is known that they are intrinsic resistant to several antibiotics, and they can also acquire additional resistant genes via different pathways. So some of these can cause disease, and that is probably why we are talking today, not only in humans, but also in animals. So in humans, they're causing mainly urinary tract infections, sepsis, endocarditis, and intra-abdominal and wound infections. So they can escape, and sometimes and can escape the gut. Now I'd like to give you an overview about the different Enterococcus species in poultry. So 7 species can cause disease and are regularly isolated in broiler type birds. So the Enterococcus cecorum is isolated from broilers and also broiler breeders. And I will cover this topic in more detail later. So it can cause sepsis, spondylitis, and femoral head osteomyelitis. Then we have Enterococcus faecalis, can also occur in broiler and broiler breeders, and mainly causing first week mortality, so navel and yolk sac infections, and also sepsis. Additionally, it's often isolated together with Escherichia coli, and it can also cause human infections. So it's, I think the most often isolated species in humans from human infections. And interestingly, there's one special disease in broiler breeders and also in laying hens. This is amyloid arthropathy. So this is a special kind of arthritis that can occur in these long-living chickens. Then we have the third species. This is Enterococcus faecium. Sometimes it can be isolated also from navel and yolk sac infection, also sepsis. And what is important here is that this is the main bacterial species that can be considered as multi-resistant and in hospital-acquired infections in humans. And 99% of these vancomycin-resistant enterococci belong to this to Enterococcus faecium. Then we have Enterococcus here. It can be also isolated from broilers and broiler breeders with sepsis, endocarditis, and also central nervous infections. Enterococcus durans, also from broilers with sepsis and also central nervous infection. And then we have two species, Avium and Gallinarum, which occur very, very rarely as disease-causing agent. So if I should rank this somehow, so according to importance in poultry, so the number one rank is Enterococcus faecalis. This is problem worldwide. The second one is probably Enterococcus faecalis because it's most often isolated after securum. More regularly, I would say, in many broiler cycles, but rarely severe outbreaks. And then we have at this ranking number 3, it's Enterococcus faecium, here durans, occasionally outbreaks, and on rank 4, the last two species, Avium and Enterococcus gallinarum, and I think they are mainly commensal. So this is a phylogenetic tree based on the core genome, so the genes that all of these species have, and you can see that there's no poultry branch or something like this in the phylogenetic tree of Enterococcus. So they are distributed more or less evenly on this and belong not to a special branch on this phylogenetic tree based on the core genome. Now I like to focus on Enterococcus faecalis. So there are some special things about this. So one thing is you can recognize from the picture here, from the blood agar plate, that the alpha hemolysis is very weak. So especially when you compare to Enterococcus faecalis, it has a very strong alpha hemolysis, is very weak in Enterococcus faecalis, and also it is more adapted to anaerobic conditions or microaerophilic conditions, so it needs reduced O2 pressure for good growth, I would say. Also, another thing which is special is that it's mainly a poultry pathogen. There are some publications about human cases, I think, but there are publications about every bacterial pathogen in humans, so there are just single cases. It's not a real zoonotic problem, I would say. And mainly the skeletal system is affected in chickens. Occasionally, we have also infection in duck. It's more sepsis-like disease there, so we have some problems also in other species, poultry species sometimes. And it's one of the most important pathogens today, as I show you in the next slides. Also, it occurs everywhere around the globe. So the impact is we have increase of mortality and especially culled chicks. So the lame animals, they have to be culled. And it's normally in the affected flocks, it's around 5 to 8% mortality, but in single cases it can be even higher. So I have heard of some flocks, they have like 30% of culled chicks. So sometimes it's very severe. Also, we have a lot of antibiotic use due to Enterococcus syndrome. We have impaired weight development in the affected houses, and we have also higher condemnation at slaughter. So this adds up to the financial problem. But it's not only the financial problem, it's also the animal welfare problems, because these animals, they have like bone infection, they cannot move their legs and it's very painful. I think so. We also on this side, it's very important disease. Here you can see a summary of the sheet you receive from the slaughterhouse, and just an example of an outbreak. And here you can see that we have like in this ECPVS affected flock, we have around 10% condemnation at the slaughter, and compared to 2.2 from the mean condemnation at the last 5 cycles. So you can see it's tremendous. And also which are the reason for the condemnation, and you see like 6% it's because of cellulitis. So it's scratched animals, and this is often seen in Enterococcus syndrome affected flocks because they cannot move so good and they are scratched, and then you have higher condemnation. And the other thing is the generalized disease findings. This is due to pericarditis and perihepatitis or sepsis-like disease condemnation. But also this can be higher. I have seen some data from like 20 to 30% in single cases. So the importance when you look into the literature, here I summed up the PubMed publication with Enterococcus syndrome in the title. And here you can see this increasing importance. And first description was done in 1983 as Streptococcus syndrome. Then we had reclassification to Enterococcus syndrome. And in 2002, the first description of bone lesion in chicken. And then in the last year, we had like, or last 2 years, we had like 12 publications. And this shows the importance, increasing importance of this pathogen. Also, there's a United States Animal Health Association broiler industry report from 2025. It's a survey of veterinarians working in broiler production and covering 80% of the total broiler production. So this is a lot. And you can see here the ranking of the main disease problems in broiler production according to these veterinarians. And you see here on rank number 4, it's bacterial osteomyelitis, kinky back. And on ranking number 6, it's Enterococcus septicemia. So yeah, you can also add this up, and you I think then you have like on ranking number 2. So it's really a problem, also in one of the biggest broiler producing countries in the world. So now I like to cover the pathogenesis. So here's an overview about this. So we have the oral uptake very early. Then we have intestinal colonization, followed by translocation from the intestine. So that means gut escape. Then we have sepsis or septic phase, and most of the time, or more or less severe skeletal phase. So during the end of the broiler production period, housing period. So starting sometimes already in week 3 to week 6 or 7, depending on how long the housing period is. So now I'd like to go into detail. The oral uptake is in the first days of the life of the broiler chickens usually. So when we working with this pathogen in an experimental setup, we inoculate a day of placement, so day 1. One oral application is enough to induce disease, and then normally 100% of the chickens are colonized. And sometimes what we found repeatedly is that the colonization decline later on. Yeah. So, for example, if you look at this data, this is data from the intestine of experimentally infected chickens, and here are the days and the age, 11 days, 18, and you see, so doesn't matter, these groups are not so important, but what you can see when we infect them at day 1, so at day 11, all of the broilers are colonized in the intestine, and day 18 also, 25 also, and then we see a decline of the colonization somehow. This is measured with a PCR in this case, so positive animals in percent. Also, this is from a outbreak which we published, field outbreak. And in this case, we sampled the broiler chickens with a PCR and from cloacal swabs. And here you can see 40 minus CT. So the higher the dot, the more Enterococcus DNA is in the swab. And you can see already at day 0, so this is normally in Germany, at least it's a day of placement. And you can see there's a group of broilers here. This one, these are below CT value 35. So that means they are clearly positive at the day of placement. So, and this is usually, they increase and like 1 week of age or even, 6 days, all broiler samples are positive, and then there's also a little bit a decline at the end, can be seen at this case again. Also concerning the colonization, so which parts of the intestine are colonized? Probably all, of course, because it's all belongs together. But what we can see here is again this Enterococcus faecorum specific PCR data and the different parts of the intestine. This is again from the field outbreak, and you can see that at the beginning, at 3 days post-hatch, all parts of the intestine are more or less equally colonized, but then later on, the colonization is mostly in the cecum with the highest colonization rates or the highest PCR values or DNA loads, you can say. And this is typical because in the ceca you have the highest bacterial concentration in general, and also of course the pathogen is called Enterococcus faecorum, and this is not by chance, so it is found in the highest concentrations in the ceca. Okay. So one additional thing concerning the translocation process is that the intestinal disease is not required. So the Enterococcus faecorum itself did not cause any kind of visible enteritis, and also other diseases like coccidiosis or Clostridium perfringens infection is not needed for the gut escape. So there was also some field work done by Luc Borst from the US. He showed that Enterococcus faecorum affected farms had no higher intestinal lesion scores, so this is coccidia scores. And also he did a co-infection experiment where he infected the broilers with coccidia, and the coccidial infection did not increase the prevalence of Enterococcus faecorum in these birds. also that is something that we find. We do not need any co-infection in our infection model to induce a disease. So the question is, how can Enterococcus translocate? So there are two ways. So the transcellular and the paracellular pathway. The paracellular pathway is important, for example, after malnutrition or in heat stress scenarios, that the tight junction proteins there are destroyed somehow or partially destroyed. And that we know. And one question is the transcellular pathway also relevant? And there's a recent publication from 2025, and they worked with a intestinal organoid model. And in this case, they infected the intestinal organoids. This is from Lonneke Vervelde from 2025. And they infected these with Enterococci and these organoid cultures. And then they killed everything outside the intestinal cells with antibiotics. And afterwards, they lose the cells of this intestinal organoids, and they plated the results. And then you can see here that Enterococcus secrum can, especially the strains isolated from outbreaks or lesions, they can survive. They go inside the cells, so they can be inside of the cells, and so it probably is both. So it's transcellular and also paracellular. So this is one very interesting work from Lonneke. And also what we found, that we also found in our research that there's a difference between strains. So not all Enterococcus secrum strains can induce disease. And I think this is very important for intervention method, that we have to concentrate on certain strains. So sepsis probably can be overlooked. So the earliest changes we can sometimes see after 1 week. We see early pericarditis lesions, or sometimes you have a slightly increased mortality. But as you can see here, this is also from a field outbreak. You see there's a little bit increased, but very, very slightly only, and you can overlook this. And especially in countries that we are in the European Union, so we cannot do prophylactic treatment somehow. So this is very important to detect this very early, not late when the bone lesions are there, because when the bone lesions are already there, it's very difficult to treat, because these animals which have already lesions, you cannot treat these. So the lesions won't disappear in the bones, of course, as you can imagine. And then you have still the losses, and you have to treat. If you treat late, you need a lot of antibiotics. So what is normal appearance, or in the field, in the broilers affected? What can you see? And as you can see here, on day 6, you already can see sometimes pericarditis or hepatitis. And then this is followed later by bone lesions. And this is an example of a field outbreak. But there you can see this shift from the pericarditis hepatitis is still there at the end, but then the bone lesions start to appear and inducing this very severe disease picture with this lameness. So why are these locations are affected? So the earliest changes can be seen around week 3, and mainly the spondylitis of the free thoracic vertebra and femoral head necrosis are seen. And these are the weight-affected location of these fast-growing broiler type birds. Of course, the broiler can't use their wings for walking, so the whole weight, it hangs on these femoral heads. And also the free thoracic vertebra, this is this one here, and you have the notarium, the fused vertebra, they are fused together, and here also, and that's why you have a lot of stress on this. So torsional and shear stress at these locations, you have microfractures in the cartilage. And if you have a pathogenic Enterococcus strain, it will go into the blood system. So it is there, and if you have these microfractures, they can be colonized, and then they growing there. The bacteria they produce can produce some enzymes which lose the bones, and also this immune reaction, the leukocytes, it induces also this lysis of the bones, and then you have this lysis of the vertebra and the femoral heads. So there's also a broiler strain influence on the pathogenesis, and this is, I think, the last slide on pathogenesis. So we did some trial with slow-growing broiler type birds, and we were able to see that less birds of these slow-growing broilers are affected. So that is maybe due to maybe not only the stress on the skeletal system. There may be some other reasons for this. For example, the immune system, which is different in these birds. Also here you can see the isolation rates from extraintestinal organs, so bones and pericardium and liver, and you see 5% of the slow-growing birds were affected, and 35% of the fast-growing ROS 308, so the conventional broiler type birds. So now I'd like to show you some pictures of the clinical presentation. Here's a septic phase, so day 12, experimental infection with retracted head, and you see it's not so well, this bird. It's like this is covered, some kind of covered posture, and so these are the birds. This in the pathology you can see this pericarditis here. It looks a little bit different some in the early stages than E. coli, so it's not so much fibrin there. Also, sometimes you see these swollen livers, sometimes brown-colored livers. Day 17 in the field from field infection here. Here you see later sepsis. So day 22 here, this bird. These are looking pretty normal, but this one closed eyes and this ruffled feathers a little bit. Also here in later older birds, you can also see more severe pericarditis, and then at the later stages, you have as a differential diagnosis, you have also E. coli, because it can look sometimes similar to it. And here you see this, there's a video I included here. I hope you can see it. Sometimes a little bit difficult with PowerPoint, but here's affected broilers. Typical the sitting position, and this bird is not moving because it cannot move anymore because it has bone lesion, and these are the typical birds which have to be culled. Yeah, these cannot move anymore. And we have also seen this in experimental infections, early lesions here, this sitting position, and this is because mainly can be because of the femoral head, but most of the time with the femoral head, they are not affected equally, both sides. So most of the time, these lesions with both legs affected is due to the spondylitis, so the infection of the vertebral column. This again, day 34. And why this is? So this is a 6 thoracic vertebra of non-affected birds. So this is a healthy one, and here you can see already from an affected one, the spondylitis. So this is somehow the swelling. And if you look and cut this sagittally, you see this infection of the vertebral body here. So there's some fibrinous capsule somehow, and here are degraded bone and pus-like material inside, and this infectious process, it compresses the spinal cord, which is here. This is not there anymore, so I pulled the spinal cord out. It's this whitish material here. Here you can see also another picture from experimental infection, and here you even can see this compression of the spinal cord, and that's why the animals cannot move their legs anymore, because of the compression of the spinal cord. These are femoral head lesions here, from field infection, from experimental infection, and also what you can notice in this case here, that's a femoral head from the outside. It's looking pretty normal, right? This is an early lesion, but you can see here, this is, and you can isolate the Enterococcus from this. So the infection occurs via the blood vessels. So most of the time, it's not arthritis. So on the femoral head is loosed from the outside, but it's loosed from the inside. And later on, it can be very severe in some cases, and the whole bone marrow of the femur can be affected. So now I like to cover the intervention method. So of course, still, and I have to say this, is that the antibiotic treatment is the most important intervention method still. Without antibiotics, it would look very, very bad. Concerning antibiotic treatment, we have a different legal situation worldwide. So in some countries, antibiotic growth promoters are still allowed. Antibiotic treatment of hatching eggs are done. So inside the hatching egg, preventive treatments can be done in some countries, in others not. And we have also differences situation about the type of antibiotics used, so due to the different license situation. So this is why you cannot compare this, and you have to also have in mind when you ask some people from different countries if they have problems with Enterococcus, you have to have in mind that probably if they say they have no problems, it's maybe due to some aspects of this what I showed you here because when they, for example, treat all the broiler chicks at 1 day of age or even the eggs, then of course they may have no problems. And additionally, if they use some of the antibiotic growth promoters, it also has an influence on the Enterococcus. So treatment should be done as early as possible. So as I already said, when lameness is already seen, it's partly effective. And I know that preventive treatment is done in regions with high pathogen pressure. And we can discuss it if it's good or bad. I think it's easy to say that it's a bad thing to do preventive antibiotic treatment. On the other hand, if you do it, you have less antibiotic use because you need very few low amount of antibiotics for this day-old birds, or in the first week you do not need high amounts, and later on you need very high amounts of antibiotics. And you have, if you're doing this, you have low mortality, less production losses, and low contamination. Of course, in the European Union it is forbidden, but we know we have the other tools are not ready, I have to say, and that is why I have to cover this, because it is done worldwide. So what are other possibilities? Non-antibiotic intervention method. So one thing is bird management. So one thing we repeatedly recognize is that in heat stress situation, we have more severe outbreaks. It is known for years that Enterococcus cause very, very severe outbreaks in heat stress scenarios. We have also seen this in houses with underfloor heating system, for example, where you have very much heat in these houses, and you have also a lot of dust because of the very dry situation, then you can have very severe outbreaks. There's also some veterinarians who advise to try to work on bird activity, that you may have less skeletal lesions when you have a prolonged dark period or something like this. I don't know if this is really that effective. In general, I would say that to avoid heat stress, it's a good thing, and not only because of Enterococcus, but because of many different things. When you have heat stress, you have intestinal leakage and more translocation, of course, not only of Enterococcus, also with other pathogens. But it's alone, it's not sufficient. So what I learned is that if you have a virulent Enterococcus faecaum strain present in the broiler house, you can do some things with management, but you will have problems nevertheless. So I think one topic that the second speaker of today will cover, and that is why I won't concentrate on this, is eliminate Enterococcus at hatchery and farm level. And that is all means also especially the breeder birds, but also the broilers, of course. And that's why I give no comment on this at this stage, I think. Feed additives, of course, is a very important tool. And concerning pre and pro and symbiotics, there's not easy to interpret results. So there's very few things published concerning Enterococcus, but there's one interesting publication from this year from Jones et al., which showed a 50% disease reduction using mainly, I think the main working ingredient was a probiotic gel applied at day of placement to the broiler chickens, and it has 2 different lactobacillus species in this, and one bacillus strain. Yeah, but there are also other reports. I think most of them are not published, but there are conference proceedings about this, that there were some other probiotics used and they had no effect at all. And even there can be that if you use, for example, prebiotics, that you have more severe disease, because prebiotics usually support lactic acid bacteria, and Enterococcus also belong to the lactic acid bacteria. So you have to be very careful with this, especially with the prebiotics, I think. Then there are, of course, other feed additives, organic acid and phytobiotics, but there's a very, no proof of effectiveness, I would say. There are only field reports, nothing really published, nothing really convincing, I would say. And but this is the situation. Then we have the bacteriophages. This is also now proof of effectiveness published. And additionally, we have a very difficult legal situation in the European Union still, because I think fixed bacteriophages cocktails are not working that good. You have to be a little bit like bacteriophage strain collection, and then or isolate from the Enterococcus the bacteriophages, and use these ones. And this is very, very difficult to license this type of intervention method. Yeah, and it's the same thing with competitive exclusion flora or competitive exclusion products is also not allowed to use them as feed additives in the European Union, because they are not defined ingredients. You have the whole microbiota inside, and that is very difficult for the licensing process. And then the thing I'm also very interested in is a vaccination. So there's no licensed vaccine available worldwide, at least to my knowledge, for Enterococcus faecaum vaccination. What we know, because of the disease, is that the protection have to be present very early. So it's probably something you would vaccinate in the breeder birds, that you can have this maternal antibodies transferred to the broilers. But it is very difficult because we have also done a research trial, and something was also done in the US from Boss et al. And they found that, and we also found that antibodies are only induce only partial protection. We don't really know why, but there are some. We can discuss it later, maybe. Yeah. But of course, vaccination is all we have to have in mind, because it belongs to the most important intervention method concerning infectious diseases. So this is something we should work on in the future also. And now I'd like to sum up. So 7 Enterococcus species can be isolated regularly from chickens, and most of them can induce disease. Enterococcus secum is the most important one, followed by Enterococcus faecalis. The disease emerged when virulent Enterococcus strains infect susceptible broiler birds. So it's depending on the strain, on the Enterococcus strain, and on the broiler genetics, how the disease looked like and how severe it is. Enterococcus secum induces mainly pericarditis, hepatitis, spondylitis, and femoral head osteomyelitis. And any intervention method should be in place as early as possible. I think this is key, because the pathogenesis starts at week 1, and we have to look at this early weeks. I think this is important. And also, alternatives to antibiotic treatment are urgently needed, and some working groups are working on this, but it's going very slowly, I would say. And then I'd like to thank you for your attention, and I'm really happy to answer your questions. Thank you.
Thank you very much, Arne, for a very nice presentation. And very informative. We have some questions here, and there is a question. Do you think chicks from very young breeder flocks, that is 24 to 28 weeks of age, that they are more susceptible to Enterococcus secorum disease than chicks from older breeders? If so, can you explain why there is a difference?
Yeah, so there are, I think, 2 things to discuss in this regard. So we know that from younger broiler breeders flocks, the chickens are more heterogeneous, and they are lighter in weight, and we know that we have some quality problems there. But on the other hand, it depends. So the thing I was thinking about is the vaccination. If you vaccinate the breeders, you will have the highest maternally derived antibodies transfer in the first weeks post vaccination. So I think this is probably not something very specific to Enterococcus. I think we have this quality problems, and if you have heterogeneous chicks, then the disease can be probably more severe. But I don't think that this is a main influence. It is probably more important is the question, where does the Enterococcus secorum strain come from? And so this can be also, so the breeders can be also a source of the pathogen. So this is probably more important, I think.
There is another one. Is there evidence that delayed feed and water intake during the first 24 hours increases the risk of Enterococcus infections later in life?
No, I think there's no evidence of this. So I think that there's nothing published, at least to my knowledge. What we've learned in the past 15 years or something is, in former times we always thought that it's important to not to feed them too early, because some people say that the yolk sac is used and it have to be used, and probably it's somehow delayed if you feed them too early. But I think that is not true, and I think probably it's a good thing to feed them early. I can imagine that it's good because you have this hatching window, and you have the birds which hatch the earliest, and these probably already have used up some of the yolk, and this is probably a good thing to give them something very early. But I don't think that this has such a high impact on. We know that hunger stress, or if we have a shortage of feed in the broiler flocks later on, it is probably more problematic due to intestinal integrity problem. I think the first days, I don't know if it's so important at the before placement, but I think if you have the possibility to give feed very early and water, it's a good thing to do. And there are some companies who are doing this already during transport, I think, and it's probably a good thing to do in general. Yeah.
There are some more questions. How do you induce infection in your animal models? And do you rely solely on the infectious doses and route of administration? Or do you also use any stressors or immunosuppressive procedure to facilitate infection?
Okay, I can answer very clearly. So the most important thing is to have a good infectious strain. And when you start with this, you have to doing first experiment, just finding the challenge strain, the suiting challenge strain. This is key, and you should work with fast-growing broilers, even if you cannot receive. You can use conventional ones, and this is no problem. You have to talk to the hatchery that they give you some good quality birds, and then you can check for Enterococcus colonization. And this is a key. And if you use a virulent strain and you have it and you see it's enough. So you can use one time oral infection with 10 to the power of 6 to 10 to the power of 8 a day of placement, and you do not need any other stressors, just this pathogen and the broiler, fast-growing broiler, and you infect one time, and then we had consistently over several tries always the same kind of disease picture with, like, it depends which numbers you have and what you want to measure. So depending on your trial design, what you want to do, but there are some parameters you can measure, for example, Enterococcus in extraintestinal organs. So, for example, this is with our strain, it's like 40%, 35 to 40%. Spondylitis, for example, it's like 20% of the birds show, or even if you do seroconversion, you have like even higher, 80 to 90%. 80% are positive then, for seroconversion. But you can also measure the weight of the broilers or something like this, body weight, spleen weight ratio or something like this. But you do not need any kind of stressors, no immunosuppressive agents, no coccidia, no wire floor model where you stress the vertebral column or something. You do not need this.
You were mentioning the differences in strains. There is a question, could it be that the disease affects Cobb strain much more than Ross?
I don't think so. I think but of course, I cannot tell you really because I haven't compared them. But I have looked into the data. I think the US working group, which are in the past, they have done these trials and they have used COP 500 sometimes. And they have sometimes a little bit more, the parameters were a little bit better even than mine, but I was not so sure. Probably it's not mainly it's also because of the strain influence, not mainly the bird. But of course, I haven't done it myself, but it's like, for example, like maybe I have like 20% spondylitis and they have 25 or something like this, 25 to 28. So it's not that.
There have come more questions that maybe also is relevant for Zoi, but you will get a final question specific for you, and that is, how do you select the bacterial strain you use in your experiment?
So it's for me, the question. How do I select? I did it this was my first experiment in broilers, and this was by chance. So I used two different strains in two concentrations, and one worked and one not. So when I would plan it again, I would use more strains. I would advise to go for 5 strains or something, to doing the very good post-mortem, to have a outbreak situation, to look for an outbreak. And if you have the possibility to do it on your own, and you have own experience in the in post-mortem analysis and bacteriology, is a good thing to have a isolate from maybe spondylitis lesion or femoral head osteomyelitis. Collect these strains and then use this. Maybe you can characterize in advance. Whole genome sequencing is not that expensive and can compare to the literature or to the published sequence. But and then you sometimes you have to go for it and to have to do a first experiment just to compare and look if the strains are working. And then afterwards you can use the one with the best results, and then you can try to answer your real research questions you want to solve.
Thank you very much. I now decide that we take a break for 5 minutes, and after the break, Zoi will give a presentation. And for those who have put in questions, I see that these questions are more related to environmental factors, and we take those questions after the presentation by Zoi. So please be back at 10:23. So, dear friends, we continue this webinar on Enterococcus infection and how to fight it. And the next presentation will be by Zoi Prentza, and she's a veterinarian and has a PhD, and she's working as a veterinarian for the Greek company PINDOS, and she's also a European College of Poultry Veterinary Science specialist, and you are a resident, so you will become a diplomate eventually or in a short time. So, Zoi, would you like to share your presentation?
Yes, hi to all of you. Just a moment.
Yeah. I can also mention that Zoi has provided lots of good practices, and they have been very important for our BroilerNet project. I can see it perfectly. You're welcome.
Okay.
Yeah, you're welcome.
Okay. Good morning, everyone. And first of all, I would like to thank BroilerNet Consortium and the organizers, of course, for giving me the opportunity to present our good practice today. And before to start, I would like just to say that was an excellent overview from Dr. Jung. The presentation was really excellent, and just to start my presentation, I don't want to remain more in details about the bacteria, what is Enterococcus, but just to to start my presentation, because in order to present our good practice with the title Advanced Biosecurity Measures for Enterococcus control, I just want to say that let's go. Maybe I don't know why I can move forward.
Is it not possible?
Okay.
Yeah.
So, okay, what is Enterococcus? We already know is a gram-positive facultative anaerobic bacteria that belonging to the family Enterococcaceae. And Enterococcus species represent an increasing concern in poultry production, and due to their clinical manifestation, potential transmission routes, antimicrobial resistance, and significant economic impact. Why does Enterococcus matter? Because Enterococcosis is an emerging bacterial disease, has become a major health and economic concern in modern poultry production. And of course, as already mentioned, there is an increasing incidence worldwide. I don't want to say more about the pathogenesis because it was really nice, all the presentation of Dr. Jung, but I just want to say only some things that is important about Enterococcus secorum. Appear to be adapted to colonize the gut from the first days of life, and whereas are commensal, we know that they are naturally inhabit the gut, the poultry gut, but some of them they can transform to opportunistic bacteria, and although there is an excellent balance in the gut, and they also contribute to microbial balance. They usually coexist with the host without causing disease, but however, this balance is really fragile. They became opportunistic, and we know it. So when the intestine, as we saw also with the presentation of Mr. Jung, they colonize the gut and is able to translocate. When the intestinal barrier is disrupted, these bacteria can translocate across the intestinal epithelium, enter the bloodstream, and spread in different organs. Yes, we know that there are dominant species and target ages, such as Enterococcus fecalis, Secorum, Faecium, Hire, and Durans, and they can cause different problems. So they can induce many conditions, such as lameness, increased culling, mortality, carcass condemnation, reduced hatchability, embryo mortality, poor chick quality, and of course, reduced performance, and important antimicrobial resistance reservoir can be. And there is also one health concern. So we have to focus on, so that's why we have to focus on this bacteria, on this disease. Okay, but we know that, as already mentioned, not all Enterococcus are bad boys. There are also good boys, but there are many predisposing factors that really can influence this condition, and this factor increase intestinal permeability and facilitate the bacterial translocation into the bloodstream. These predisposing factors are the rapid growth, skeletal immaturity, the stress, environmental stress, gut dysbiosis, intestinal barrier damage, some intestinal diseases such as coccidiosis, necrotic enteritis, poor water hygiene, wet litter, heat stress, high stocking density, mycotoxins, poor biosecurity, pathogenic clonal strains. So we have to think about it. The problem is that Enterococcus can persist in house environments and has a strong ability to form biofilms. And even more, can transmit through water. The surfaces can remain in the surfaces through the dirty eggs, and can negatively affect the gut health and the performance, of course. So the critical question becomes, how can we break the infection cycle? This led to us to develop this good practice based on advanced biosecurity measures for Enterococcus control that are all these measures in order to interrupt the infection cycle before clinical disease develops. So our good practice is based on 3 pillars. Number one is environmental control and structural integrity. Water, the second one, water hygiene and biofilm control. And the third one, targeted on microbial management and gut health. The core concept of this good practice is to prevent the colonization instead of treating the infection. So the first pillar focuses on eliminating environmental reservoirs where bacteria can survive and multiply. It's nice to say that the farm design plays a crucial role in biosecurity. So non-porous surfaces, floors and walls, the smooth and washable infrastructure, reduced bacteria persistence, and of course improve the disinfectant efficiency. The goal of this is to eliminate the environmental the biofilm from the environmental, and that can support the bacterial survival and growth, of course. Infrastructure alone, however, is not sufficient, and it's important to design and implement an effective cleaning protocol that may last approximately 3-5 days. It depends, of course. This begins with dry cleaning, washing, hot water application, disinfection, and complete drying. The key effect is the biofilm, of course, disruption, increased disinfectant penetration, and prevention of pathogen survival. If we have some outbreak, we can focus more and insist more with the cleaning and disinfection. So intensify the cleaning procedures and maybe extend downtime between the flocks. This is important to break the infection cycle completely. Why cleaning and disinfection, I would say, matters? Because we have to avoid the generation that the biofilm form, and this is this is very important because, as we know, after the disinfection, cleaning and disinfection, the total flora in that we have in the surfaces inside the building may decrease. But when we have unoccupied periods before the animal, of course, arrive in the house, immediately after cleaning and disinfection, this flora can increase. So it is very important that to know that biofilm can protect bacteria from disinfectants and can remain some bacteria inside, of course, the buildings, and can persist in the environmental. It can also survive until the next flock. So the key message is to eliminate biofilm, and in order to have no persistent contamination. Of course, we know that biosecurity, hygiene, and biosecurity standards are essential, and in order also to control the traffic in the farms, we have to focus on this picture, and also in every stage. So we have to think in all these important things, all these factors that can broke the normal situation during the production cycle. So the second pillar is the water system hygiene. And I would say that is very important. Water is a major route of Enterococcus transmission. Contaminated water can spread Enterococcus, and biofilm can form inside the drinking water lines. And what we have to do, of course, to focus on this, to clean and disinfect also the water lines, using thermal disinfection. Of course, hot water flushing is really important. The water line cleaning, and in order to remove the biofilm, to eliminate the biofilm. Another thing is it's important to regularly test the drinking water for bacteria. This helps to detect the contamination early. So if we test the drinking water for bacteria, we can act very early. We can think what we have to do, and in order to have a safe, clean water supply. The third pillar is targeted on microbial control, and it is a different philosophy. That this concept is based on microbial competition, and of course, instead of focus on eliminating the bacteria, it aims also to establish a beneficial microbial environmental that naturally limits the pathogen colonization. In this context, the environmental probiotic application is essential. This can be after cleaning and disinfection in order to create a positive biofilm. It can apply after the complete drying, creates a protective microbial layer, and supports the early life gut colonization. So the important is that chicks placed the following day for maximum effect. So we don't have to wait a long time before placement. Prevention of vertical transmission. I would say that this is really important. So we have to think the same thing for all the production stages. So either in the broiler breeder farm, either in the hatchery, and in the broiler houses. So we have to focus on hygiene and high biosecurity standards in all these stages. Of course, in a hatchery, it's important to separate dirty eggs from clean eggs. Also to know that if we want to eliminate the floor eggs, and of course the reduction of vertical pathogen transmission, because these bacteria can exist also in the broiler breeder farms flocks. The goal is to ensure chicks start life in a low pathogen environmental. About the gut microbiome stability, it is the same concept, but we want to have a healthy microbiome in the birds, in the gut, poultry gut, in order to protect the birds. So we have to promote microbial diversity, enhance the colonization resistance, and of course limit the pathogen establishment. The concept here is the competitive exclusion of Enterococcus. We can also use some in the flock, of course, some gut health promoters to support gut health, such as probiotics, symbiotics, other phytogenic compounds. Some examples here: thyme, oregano, garlic, mint. And that can have that can improve the gut integrity, help to balance the microbiota, and of course to reduce the pathogen pressure. An integrated approach is required across all the production stages in order to have healthy birds and nice performance. So we have to think that prevention of Enterococcus colonization at all levels. It is really, really important. The key message to take home is the prevention over treatment, elimination of environmental reservoirs, of course, biofilm, control of waterborne transmission. Biofilm disruption is essential. Stable gut microbiome ensure the resistance, and improved flock health and productivity in order to have a healthy environmental, healthy microbiome, and healthy birds. This is really important. About the economical benefits, of course, that it are attributed on reduced mortality, improved FCR, better welfare, and lower medication costs. About the future, about the future, looking ahead, several emerging technologies may further improve the prevention of Enterococcus, like whole genome sequencing. Of course, will enhance the understanding of bacterial epidemiology and transmission pathways. The identification maybe of virulence markers may facilitate earlier detection of highly pathogenic strains. Maybe the research in vaccination, we don't know if it works really. Microbiome engineering and innovative technologies such as photocatalytic disinfection also offer promising opportunities for future disease control. Thank you.
Thank you very much, Zoi. Very nice, interesting presentation. We have some questions here, and I start with those that are maybe more specific for Zoi. And Arne, please join if you want to comment on this. There is a question about which disinfectants have been shown to be the most effective against Enterococcus faecarum, and are there any active ingredients you particularly would recommend? So Zoi and Arne, if he wants to add something.
Okay, you can. I think that all may have their effectiveness, so it depends. And I don't want to stay only in one because really we use many also here. And I think I would say we have to change. Okay, and in different flock, maybe it's better to change the disinfectants.
Arne, please.
Yes. So,
And sorry, just one thing. I think that it is better to have very clean, very, very clean surface, and then maybe we will not have problems with the disinfectants.
Yes, I think I can support most of these. So we have done one study concerning the environmental survival of Enterococcus secorum, and also at the moment we are doing a project where we test the susceptibility of Enterococcus secorum to different disinfectants. And I think one general thing is that you can see some lower susceptibility in certain strains, but most of the time these are, I think, at lower concentrations. I think the key point is that most of the disinfectants probably will work if you do the disinfection procedure, as Zoe told you already, is properly done. I think the main problems is not the substance as itself, but more the other factors, as you have a broiler house which cannot be disinfected because it's the floor and the is have big cracks and there's a high protein contamination still present and so on because it's not properly cleaned, or you have the wrong concentration of the disinfectants because your diluting device it's not or dosing device is not working properly and nobody checked it ever and such things. I think this is there are several things you cannot disinfect properly because when you spray inside it will go broke. For example, there are technical devices or things that are very difficult to disinfect because you have to use foam because everything that is up in the houses at the ceilings and so it's very difficult to disinfect a broiler house, I think, and this is a main problem, not the disinfectants itself. It's my impression.
I agree with you.
Yeah, and I'm curious. I have no question here, and that is, I know in some countries they are used like soil floor, so it's not concrete floor. Is that a risk factor, or it doesn't matter if it's a concrete floor or a soil floor?
I think the housing practices in broilers, they are different around the world, and there are many differences. So, for example, in the US, they are often have no concrete floors, but the type of the floor is not the key point, but the key point is that they use the litter in several broiler cycles after another, the same litter, and you have like a competitive exclusion effect because you have all the bacteria present. It can be good, it can be bad. It depends what you want. But of course, if you are doing this and you have, and I think there are many influences, of course, also on Enterococcus. If you have different management practices, so if you have different management practices as this has a very big influence. It is not the type of the floor, but it's more like this competitive exclusion effect. I think this is the biggest influence at this point when you ask this type of questions. But yeah, and also you have of course influencing factors as the downtime in between the cycles, the length of the times, and thinning, which is used in Europe but not in the US, and so on, and the different types of feeds, and so it's a lot, and all have influence, of course, on the disease.
Zoe, please, do you want to comment on?
Yes,
floor?
I agree.
Yeah.
I agree with you, Dr. Jung. I think it's the effectiveness of all these disinfectant are remain on what we have to do before this. So here in Greece, we started to build farms with I don't know if I'm saying well, the clean line. So well designed with smooth surfaces. So it's a really, really excellent way to just with only water, you can remove everything, the dirty things inside. So it's really an excellent way to clean and after to disinfect very easy and very quickly. Regarding the use of the soil, like a floor for the like a litter, I don't know. Here I never seen this, but I think that okay, we will have many bacteria there, but we have to know that there is a competition, competitive exclusion, but we don't know exactly how many are the pathogens and how many are the good one.
Mhm.
So I don't think that it's a good way to risk about this, and it's good to maintain the procedures. I think the biosecurity in all the context.
There is another question, and do you think that high pH in the water could improve clinical signs? I think it's reduced the clinical signs probably.
About some acidifiers in order to decrease the HP.
Yeah.
Yes, I think okay. But first of all, it's a good way to not skip the normal stages. So if we have water lines, the biofilm inside, I don't think that acidifiers can do miracles.
Mhm.
So first of all, we have, in my opinion, to check, to test the water, and then to act. And of course, we can test also about the HP. Okay, we know that it's better to have a low HP.
Arne, do you have a comment on this? On the pH of the
Mm.
water that could improve the situation?
Um, mm, I support Zoe in this that you have to have the clean water lines in advance, and then you can do something. You can many broiler farmers are using acidifiers during the cycles, and it lowers the bacterial amount in the water. That is clear, and I think it may have also a positive effect, but I don't know. So for me, when we talk about this whole thing, it's a little bit sometimes the farmer can do everything, but if the pathogen is introduced via the day-old chicks, it's difficult for him, right? Because they are already colonized, and you can do everything afterwards, but it won't help that much. So I think we also have to focus on this part, on this transmission process more in the future, I think. And it is also a little bit delicate, of course, and difficult to discuss, because we don't want to point with a finger on some parts of the production chain, but it's more like solving this together, I would say. More like this. But if we do not talk about this, then it will be difficult, I think.
There is another question on, in your experience, is Enterococcus primarily a gut health problem, or do environmental factors such as ventilation, ammonia, carbon dioxide, humidity, litter quality play an equally important role in determining whether the bacterium becomes pathogenic? It has partly been answered here, but Zoi, would you like to comment on that?
Of course, as we already mentioned, both of us, predisposing factors. So the environmental, the microclimate, the bad condition inside can predispose the infection. And as we know, not all the bacteria, not all the strains are bad bacteria, but they can become opportunistic. So we have to have an holistic approach on this. So yes, we have to think that if we care about all the stages, all these things, nutrition, cleaning, disinfection, the growth, all the condition in the flock, the management, I think that prevention is better to treat. Yeah.
Arne, would you like to comment on that?
Yeah, so because of my experience, my own experience with the animal trials, which I have done, so I have a little bit more of a pathogen concentrated view on this. So in my opinion, it's more a question if a pathogenic strain is present or not. And if it is present, then it's very difficult to do anything about it. So the factor, because most of us and most of the farmers, we have this fast growing broiler bird is the most effective bird, and it's used in 99.9% in the world broiler production. So this is the birds we use, and these are very susceptible to these pathogenic strains, and we have to think about this I don't think that you can do anything if you have these strains present in your flock. Then at the moment you have to treat, and nothing can be done about this if it's present. So the question is, what is very difficult to answer, how these strains evolve, and if this can happen in a broiler flock. It's very difficult to answer this if it's really possible. So, because what I'm thinking is that as if infection takes place so early, it's probably not the case that in each broiler flock you have this transition from pathogenic from commensal to pathogenic. I don't think so. So probably it's induced somehow, or it is still on the farm. And so there are both cases. So I think that is why it is also very important to clean very thoroughly and to disinfect. And because we have also seen that in some farms we have the repeated outbreaks in single houses. And so this points to this, that is not this one way of epidemiological introduction. So you have not this, you can have both. It can be stay on the farm, or it can be introduced newly. So that is why we have to cover all aspects.
There is a final question. I know that we are running over time a little, but this is the final question, and it might not be so easy. In the field, how can you distinguish between an E. coli infection and an Enterococcus cecorum infection before we receive the laboratory results? Zoi, do you have an idea on if this is possible?
Okay. Not always is possible, but in my experience, when we have skeletal disorders, I think there will be Enterococcus. And in addition to Enterococcus, we can find Staphylococcus, we can find an E. coli. But when we have, and I don't know if you can see in my presentation, some pictures from our field situation. Sometimes we can also find, just one moment. this and this. So in this case, I can say 100%, but we are quite sure that we have to do with Enterococcus.
Yeah.
Okay, so
yeah, please.
I think this is difficult. I don't know if this question makes so much sense. It's because we have to do also diagnosis. But of course, if you have this classical first week mortality problem, the early E. coli infection, most of the time you do not see this in the Enterococcus secorum infection. So this early mortality peak is very seldomly seen. It's more like this skeletal form later on. But of course, there are also some flocks which show classical arthritis infection, and then in combination with femoral head osteomyelitis and so on, and there you cannot differentiate. You have also the E. coli staining, which can induce arthritis. So in some cases, it may be difficult to just see to diagnose from the clinical picture. It's not possible. And also what Zoi says was with this even the bone lesion, even if you have spondylitis, sometimes we isolate other bacteria, of course. It's not specific for Enterococcus secorum. So you can isolate E. coli, Staphylococcus aureus, sometimes Enterococcus faecalis. But to my knowledge and what we have seen in the field is that the most severe outbreaks, this is caused by secorum. Yeah.
Thank you very much. And that will be the final thing. It's a very interesting and important topic. I would like to just in the chat share with you the information sites you can find about BroilerNet. And I will just have the final words here, and that is that BroilerNet is soon to be finished. However, our material, as this good practice, you can find the fact sheet by Zoi in our BroilerNet Knowledge Hub. And I put in the chat the link to this website. And I also would like to acknowledge our BroilerNet Work Package Leader team. And in particular, I would like to acknowledge, Professor Natalia Mayo-Masferrer, who was organizing this topic. And thank you very much. And thank you very much, Arne and Zoi, for giving very nice and interesting presentation. I learned a lot. I hope that this knowledge is spread, and we have, I think it was about 40 people online. And then it will also be a video that will be available at the BroilerNet site in future. It will come in a couple of weeks. But if you want to have look at it again, it's possible. And by that, I hereby will close this webinar. And thank you very much for joining us. Have a good day and a good summer. And thank you very much.
Thank you.
Yeah, thank you, Zoi, and thank you, Arne.
Yeah.
Really interesting. Yeah. Thank you very much.
It was a pleasure.
Thank you. Bye.
Bye-bye.
Bye-bye.
Bye-bye.